Hi, everyone. Thanks, Dylan, for that introduction. Uh, this is the first time I have ever presented to a group via Zoom. Usually I'm able to see your faces and ask you questions about what you know, but I can't do that tonight too easily. So I'm going to assume that you know nothing. And we'll talk a lot about basic lichen biology and their structure and their lifestyle, how they earn a living, how they reproduce. We'll talk about what roles they play in the environment and how they fit in and otherwise react with other things that are living with them. I've included a section that we can have some fun with on the challenges that the lichen symbiosis pose to our concept of what it means to be an organism. We'll also spend some time on what it takes to tell lichens apart from each other. Now, this is not a class on lichen identification. I don't expect you to remember any of these things. I do hope that you can look at the pictures, see some of these structures and say, wow, there's a lot more to these things than I've been paying attention to up to this point. And they are worth exploring. And I can guarantee you that they are worth exploring. They're mostly small things and you need to look at them through a hand lens. And that's one thing I would hope that if any of you come to the walk on Friday, is that you bring a hand lens if you own one. Other names for them are jeweler's loops, that kind of thing. A good magnifying glass if you don't have a hand lens. And I'll teach you how to use it properly so that you can get the most out of them. In the event, we'll finish with a section titled 10 Common Lichens in Henry Cowell. And I chose those lichens completely on the basis of which ones we will see on Friday. So we'll start out with kind of asking what a lichen is. And before we do, I want to point out this painting by this guy named Ernst Haeckel. If you've never heard of him before, Google him and look at this. He had a book named Art and Nature that was published in 1904, and it has 100 paintings of various groups of organisms. It really is a wonderful book. He painted bryophytes and lichens and squid and starfish and snails and almost anything you can imagine, and really did wonderful job painting them. So what are these things anyhow? Some of you probably know or have heard that lichens are a symbiotic relationship between a fungus and an alga. Now, I don't want to assume that you really know what a fungus is. It's something that we all think of, oh, they rot logs, don't they? Or they spoil my bread or my orange. But that's only the very outside of their roles in nature. They are a kingdom to themselves. They're not a plant, they're not an animal. They are more closely related to animals than they are to plants. They cannot produce their own food like plants can. And that's the first and most important thing you need to know about a fungus when it comes to lichens. So the idea with lichens is that the algae, which is called the photobiont, photosynthesize and they create sugars, and the fungus, which is called the mycobiont, uses the sugar as food. And they do this with this basic building block of fungi, which are strands of cells. And I want to say from the get-go that fungi cells have nuclei like our cells do, and like plant cells do. But these strands wrap around the algal cells, as shown in that little inset picture to the right, and either just touch the cell wall of the algae or penetrate it, but they don't penetrate the cell membrane itself. In the meantime, these sugars are being transferred to the fungus through this little penetration body called a haustorium. And if you think that this is just, well, we grow around that, you have to know that as the lichen develops, the algae and the haustoria are actually developing together and modifying each other as the lichen grows. Now, the algal component, which we remember called the photobiont, can be a green algae, or it can be a cyanobacteria, which is a blue-green algae. The difference between the two is cyanobacteria have no nucleus in the cell. They both photosynthesize. Blue-green algae can do another good trick, though, which is to fix nitrogen. Nitrogen fixation is a process that all plants need because nitrogen in its atmospheric form cannot be used by plants. It has to be added to either hydrogen or oxygen, and then the plants can take it and use it in their cells. So this nitrogen fixation is a very important function. You can often see or tell which kind of photobiont a lichen has by the color of it. Black often indicates, but not always, but often indicates the cyanobacteria, whereas lighter colors are virtually always green algal. And if you break a lichen, if it's a large lichen, you can break it and actually see the algal layer with a good hand lens and see the color of it. If it's bright green, you have a green algae. And if it's dark blue-green, you're looking at a cyanobacterial photobiont. Well, let's not go too far without just saying these things are spectacularly beautiful. And I think they're one of nature's best art forms. You have to put them right up there with tropical birds and any flower that you can imagine. And I would just want to have a little section here where I'm not going to say much. All of these lichens are like local lichens that I'm going to show you. And just appreciate the forms and colors and shapes that lichens can take. Welcome to the Amazement Park. So let's just take off the adult glasses. I see most of us are adults in this room. And let's look at these things with the eyes of a child. Now, if you were standing with me, I'd be hearing oohs and ahs at this point. So I hope that I'm imagining that you're saying those things.
Ooh.
Thank you. Oh boy, these colors and shapes, they're just, I just can't get over them. It's why I'm so obsessed with them now. So let's start putting some names on these shapes. Lichens are generally described as one of three forms. Fruticose is generally three-dimensional and usually round in cross-section, but sometimes flattened a little bit, but they stand up from the substrate in general. The next one is foliose, and that's generally flat. It has an upper and a lower surface that has some difference in appearance, but these are not strongly attached to the substrate. If you wet them a little bit and get a little knife blade or a credit card underneath them, you can generally remove them from the substrate fairly easily and look at the lower surface. The third most commonly ascribed form is crustose, and that's also flat, but it adheres so strongly to the substrate that you can't separate it without either taking some substrate with you or ruining the lichen. So when I collect these, if they're on rock, I have to chip away rock, or if they're on bark, I have to carve some of the bark off of the surface so that I can put the lichen in a packet and bring it home and identify it. But wait, there's more forms, and these can generally be shoehorned back into one of those other three forms. But I like talking about pin lichens, and we'll talk a little bit more about them later. These are fun guys to find. They're fairly rare and very small, and it's easy to walk right past them without seeing them at all. And the last are the dusts. These are nothing but a tiny powder on the substrate. You can see these very commonly on redwood trees, a bright sea green color, or sometimes a blue-green color on the redwood trees in Henry Cowell. And these are nothing more than a few strands of fungus wrapped around a few algal cells, as in this little insect picture down below. They're also common on road banks, on whether they're dry or moist. But the dusts, I don't believe we'll see any. Oh, yes, we will see one on Friday. So let's look at what happens if you cut one of these things in a section. Let's start with the foliose lichens on the bottom. Remember, there's an upper and lower surface that's somewhat differentiated. So they'll have an upper layer that's strictly fungal, and that is called the cortex. And directly below that is the algal layer. This is where the algae are living and photosynthesizing, and where the haustoria are connecting to the algae and getting the sugars from it. Below that is the medulla, which is usually white, but sometimes other colors. That's also strictly fungal. And the lower cortex is also fungal. And then on the bottom are some bodies called rhizanes that are often present on foliose lichens. And those are attachment structures. They're not roots. They're not hollow. Nutrients are not being drawn up from the substrate into the lichen. There's simply attachment. Now, on the fruticose side, on the left, we also have the outer cortex and the algal layer directly below it, because these are three-dimensional, there can be no lower cortex. And so we then have the medulla being the central area of the lichen. And again, the cortex and the medulla are fungal. The only place the algae are living are in that algal layer. Well, let's talk about how these things were discovered and how they were first described. In 1867, a guy named Schwendener in Switzerland first decided, or first discovered, or first hypothesized that they were a relationship between a fungus and an alga. That's 1867. About 10 or 12 years later, a couple of German guys coined the term symbiosis, living together. And that term was coined because just to describe lichens. Now, Schwendener's idea was kind of scoffed at first, and it really wasn't until World War II that it was generally accepted by the scientific by scientists. But since then, it has stood as scientific gospel. But in 2016, a guy in Montana named Tony Scribbly discovered that most lichens also have a yeast embedded in the upper cortex. And subsequently, it's been shown that wolf lichens, which is the chartreuse green lichen that you'll see in the Sierra when you get up into the higher country on the trunks of the trees, those have two species of yeast growing in the cortex. Now, what is a yeast? A yeast is simply a single-celled fungus. It's in a different group of fungi than the ones that form most of the lichens, and we'll talk a little bit more about that later. But what does this mean? Everything that everybody's thought about and investigated about lichens has to be revisited now. What role does this yeast play in lichen formation, in chemical production? How do these yeasts earn their living? Are they the glue that allows the fungus and the algae to create a lichen? But I think what's really important about this thing is to remember that science is an ongoing process. It is not truth itself. It's simply a process by which truth is discovered and described. So remember that science is always asking the questions. Did the last guy get it right? Can we improve upon our knowledge? So where do they grow? Well, in a simple phrase, everywhere. They grow on all the continents. They grow on rock. They certainly grow on trees. They grow on bryophytes. They can grow underwater. There are species that grow underwater. There are species of lichens that grow on barnacles in the intertidal zone. And we believe that we just discovered a new species down in Younger's Lagoon, growing in the intertidal zone. They also grow on painted metal mailboxes and cars and leaves. They grow on leaves or pine needles. Now here's a picture. This is an extreme close-up. You can see along, here's the midrib of a tan bark oak leaf, and here's a lichen growing on the surface of that leaf. So they've pretty much found a way to grow on anything. So let's think about how to think about this lichen symbiosis. What is it going to tell us about itself and ourselves? I believe, and I like to put forth the idea that lichens are an organism in themselves. They're beyond a symbiosis. Now, note, and I haven't told you this, but the fungus without the algae don't look anything like the lichens that they can turn into. Now let's look at the algae for a minute. In each cell of every cell of every green plant has bodies inside called mitochondria and chloroplasts. The chloroplasts are where photosynthesis is happening. And the mitochondria is the place where the sugars that the chloroplasts are producing are being used and turned into energy so that the rest of the cell can use that energy. But something that is really interesting about both of those organelles is that they were once separate organisms in their own right, and they became absorbed into the protoplasm of some algal ancestor, who knows where that was. And that organism said, wow, these are pretty cool things. I can all of a sudden, if I don't eat and dissolve these things, I can now make my own food and I can use it. The DNA in mitochondria, the DNA in the chloroplast, and the DNA in the plant cell nucleus, all of that DNA is different. So every plant cell is a compound organism in its own right. Now, consider your own body. I'm going to keep making this argument that lichens are an organism in their own rights. More than half of our cells are not human. There are more bacteria than human cells in our body. And most of these bacteria are critical to our good health. We all know about the gut flora and probiotics and how without those things, we're sick puppies. There's also a whole flora of bacteria and viruses and fungi present on our skin, and a good number of them are helping protect us from the bad guys. So I would like to put forth this idea that lichens are a model for all of us to think about what an organism is. If we are compound organisms, if all plant cells are compound organisms, why can't we just think of lichens as a single being? But we can play with this a little bit more. Lichens, we can also think about them as agriculture that were invented by fungi. That's a common one. If you turn that one around on its head, you could call a lichen a gall, like the apple galls on oak trees, that the algal cells make the fungus create. If you really want to get back to it, you can say the fungus is not doing anything good and it's just stealing sugar, so it's a parasite. Or maybe it's an algal greenhouse that the algae has said, like, hey, you build this for me and we can live up in the trees and I can photosynthesize. But more than that, you can also look at it as an ecosystem in itself. So we have an ecosystem has to have producers, which in our case is the algae, and it has to have consumers, which in our case is the fungus. We also have associated bacteria and algae and fungi growing on the surface of the lichens, and these are called parasymbionts. And you think they're just having a surface to live on? No, some compounds created by these parasymbionts have been shown to be beneficial to the lichen so that they taste bad to things that would eat the lichen, that prevent other fungi from eating the lichen. They can provide vitamins to the lichen. So these parasymbionts are part of this same community that you would say leads us to say that they are an ecosystem in themselves. Now, this symbiotic concept probably evolved before plants colonized land. Remember, since fungi can't produce their own food, they had to eat something else. But these guys found a way to do that without killing their hosts. And that's pretty nifty. And if you start studying fungi at any length, you'll find out that they have evolved to make a living in just every way you can imagine. Now, this evolutionary building block has given rise to lichens and also to these mycorrhizal associations that have come to be so popular in common science today that forests cannot live without. Now, lichens have evolved in 7 different fungal lineages. And what I mean by fungal lineages, remember we started out by saying that fungi are their own kingdom. And so inside that kingdom, there are subdivisions, and lichens have evolved 7 different times in those different subdivisions. Now, one of the peculiar things is that the names of lichens are based only on the fungal partner. I think this is silly, but that's the way taxonomists and lichenologists have set it up. The reason I think this is silly, for one reason, I think, as I said before, I think they're organisms in their own right, but the same fungus can make a different looking lichen with a different alga. And those lichens will look completely different. And how do you put a name to that? You're going to call it the same thing. I think someday somebody will say, we're going to change that method of taxonomy. We'll use all these old names, but we're going to start giving new names to something that has the same fungus in it. Not anytime soon, though. So, how do lichens reproduce? Lichens use both sexual and asexual means of reproduction. So let's look at the sexual side first. Fungi are the only ones of the partnerships that use sexual reproduction. algae their cells just split. This algae that are in lichens, their cells just split. But in fungi, they can reproduce sexually on their own. And in this situation, the fungi are only reproducing the fungus. They're not reproducing the algae. We'll talk a little bit more about what that means, but let's look at this. Either two of these fungal strands, which are called hyphae, can meet and conjoin, and they'll form a cell that has two nuclei in it. That's one method. Another method is these asexual spores can be formed, and they can land on this little thing called a trichagon. And again, that cell with two nuclei stays around for a little bit. At some point, those two nuclei fuse and a zygote is formed. And then we go through the process of meiosis, and eventually you end up with spores with only one set of chromosomes. The hyphae are not male or female. You can't think of them that way. They are only mating types. Luckily, in the group of fungi that produce most of the lichens, there are only two mating types. But in mushrooms, for instance, there can be as many as 2,800 mating types. And the permutations and combinations can be extremely confusing. But the idea is that they can only mate or fuse with a mating type that's not their own. So after the nuclei fuse, spores are formed in a sac called an ascus. And this body, this matures in a body that's either atop of the thallus, which is called an apothecium, or buried in the thallus, which is a perithecium. Now, asci, which is the plural of ascus, is one major group of fungi. The other are basidiomycetes, and their spores form on these little club-shaped structures. Vast majority of mushrooms are basidiomycetes. The vast majority of lichens are ascomycetes and have their spores created in ascus. So when that spore is released from the ascus and lands on an appropriate surface, it can be carried by an animal or the wind. It germinates and it needs to find an algal cell. Remember, this is the fungus reproducing only. If they fail to do that, they will soon die. Now, without the algae and maybe the yeast, we don't know, the fungus doesn't look anything like a lichen. Now, I would think that finding an algal cell on a substrate is probably pretty hard, but given the abundance of lichens, it's obviously not that hard. And we can probably even see on Friday when we take our walk, we'll see on tree bark or on rocks, there's algae growing on it. And so lichens have taken advantage of that, and they can form the thallus. So asexual reproduction kind of gets around that difficulty, and it uses two different specialized body called Isidia and Ceridia, and they both carry the fungus and the algae. And so let's look at the Ceridia first. The Ceridium is just an algal body, just an algal cell or two with a bunch of fungal hyphae wrapped around it, and it forms this little powder-like thing that you can see here on the edge of this macrolichen. And those Ceridia can be brushed off again by the wind, by any animal that comes in contact with them, by insects, and carried to a new substrate where they can grow into a new lichen thallus. Isidia are basically the same thing, except they have a cortex And again, this time it breaks off from the surface of the lichen and lands on a substrate. And they're starting over with the algae and the fungus present, and they don't have to look for each other. So those two bodies are the main ways that lichens reproduce asexually. So what role do lichens play in the environment? And this is probably the question you're going to get a lot from your park visitors. They play a number of roles, and since they grow on almost every surface has its own, lichens had their own job to do in that situation. Lichens are colonizers on substrates that are not available to vascular plants. What does this mean? Well, colonizers have to be stress tolerant. What happens in California when a lichen is growing on a rock or even a tree trunk, and it's the middle of July and it hasn't rained in a month and a half, and that rock is dry or that tree bark is dry? Well, lichens have to be able to handle that situation. They have to be able to tolerate that desiccation. And so they are what I like to call cold-watered, like we use the term warm-blooded or cold-blooded for different vertebrates. There's a fancy scientific word called poikilohydric, which means their water content equalizes with the water content of their immediate surroundings and substrate. Now, not much is known about the mechanisms of desiccation tolerance in lichens. A lot more is known in bryophytes. But in both groups, the bryophytes and the lichens, there are chemicals and compounds that make that dry lichen UV resistant so that the algal cells don't get damaged by UV light while they're in this dormant stage. So while they're colonizing this substrates, they're helping to create soil. And they do this in a number of ways. The lichen acids dissolve rock to a certain extent. And in some cases, the fungal hyphae actually penetrate the rock. In fact, there are some lichens where you can't see anything but the apothecia on the surface, and the algae and the lichen, I'd say the fungal partner is inside the rock. And as the lichen is growing on whatever surface it is, the salus is trapping other detritus, and this builds up biomass to the point where primitive soils are formed, and you can start to see small vascular plants growing. Another important role, and this is particularly important in the Pacific Northwest, is nitrogen fixation. Remember, we talked about the cyanobiont being able to fix nitrogen so that plants can use it. This lichen, which is called lobaria, is extremely common in the Pacific Northwest. It can have about as much as 1 ton of biomass per hectare. And since it has a blue-green photobiont, it fixes nitrogen and contributes 50% of the nitrogen needed in the Douglas fir forests in Oregon. Why does it look green if it's got a cyanobiont? Well, this is a picture of the lichen when it's wet, and everything looks green when they're wet. Lichens are also nutrient traps. What does this mean? Well, consider for a minute how fast it takes any horizontal surface in your house to become covered with dust. The air is full of dust and nutrients, and these nutrients can be caught by these pendant lichens. Also, other macrolichens can catch these nutrients, but the pendant lichens are really, really good at it. And these nutrients can be eventually delivered to the roots of the tree as rain washes them out, or as they fall off and land on the ground and rot. The lace lichen, which we'll see on Friday, has been shown to contribute 4 kilograms per hectare, which is 3.5 pounds an acre, of nitrogen to oak forests. And this nitrogen is not coming because they're fixing the nitrogen. They don't have cyanobacteria. This is being caught from the atmosphere. So a really, really important point, because this is a very commonly held myth that lichens kill trees. They are actually beneficial to trees, and many studies have shown this. Why are they growing on dead trees so much? Everybody says, oh, they're growing on dead trees, they must have killed it. The exact opposite is true, but the reason they're growing on dead trees is there's plenty of light, and they're opportunists, and it's easy to photosynthesis without leaves in their way. So this is a really important myth, because people say, oh, I have to kill the lichens, they're going to kill my trees. No, the exact opposite is true. Well, lichens are, as we discussed earlier, they're communities in their own right. And not just the things growing on their surface are involved with lichens, but arthropods, nematode worms, tardigrades and rotifers, all tiny little critters, are living amongst the lichens. I never look at a lichen under a microscope after I've collected it without seeing something walking around. Also, we talked about the parasymbionts growing on the lichen surface. Another thing we have to know is that some fungi parasitize other lichens. They will either eat all of the sugars and kill the lichen. Sometimes they will steal the algae from the other lichen and become the parasites in the beginning of their life cycle, and then become independent after they've stolen the algae. So, as I said, fungi have figured out all sorts of different ways to earn a living. Animals use lichens. We know that in the tundra, caribou eat them. And here in California, slugs can eat them. Hummingbirds in particular use lichens as nesting material, a lot of times for camouflage. But I have seen lichens in other birds' nests, usually fruticose lichens, as nest linings. And insects have co-evolved with lichens with these spectacular camouflages. And there's a moth on the left. We have a moth similar to this in the Central Coast, but you can barely see it. And on the right is a caterpillar that I found in Calaveras County, and it's almost invisible. I was really, really lucky, but the reason I see it is because I walk around with a hand lens all the time and I'm looking at lichens, and I came across this thing and I said, oh my God. Well, humans use lichens as well. In Europe, there are some that are used as the basis of perfumes. A lot of people around here use lichens for dyeing. Some really wonderful colors can be created with lichen dyes. Scientists use lichens as indicators of air pollution. And because those lichens absorb their nutrients from the air, they also take in pollutants, and the algae can be sensitive to those pollutants. And if they are toxic to the algae, they'll stop photosynthesizing, and then the lichen will die. Some lichens are more sensitive than others, and so their presence indicates how clean or dirty the air is. And there are tables indicating the maximum level that any particular lichen can tolerate. And there are tables for nitrogen, sulfur dioxide, acid deposition, ozone, fluorine, heavy metals, you name it. If you're interested in this, there's a book called Macrolichens of the Pacific Northwest, which I'll show you a little bit later, by McCune and Geyser. And they have some of these tables that you can tell how clean your air is. So let's get into a section of how do we tell lichens apart. There are more than 1,800 lichens in California. I have no idea how many lichens there are in Santa Cruz County or in Henry Cowell. I'm working on that project, but it's going to take a long time. But I don't want this to be a lichen identification class. I want you to look at this section as the ways in which lichens have evolved and all the different forms and structures they've evolved into, and then we can use those structures to identify them. So what do we do? What do I do when I bring a lichen home? Well, growth form is a good place to start. I do have to say that any given genus of lichens can have fruticose forms and crustose forms and foliose forms. So they're not taxonomically, I don't want to say they're not important, but you can't rely solely on what form they are. But it's a good place to start. What is that lichen growing on? What substrate is it growing on? Most lichens have a preference between substrates, whether it's on rock or on tree bark, on rotting wood in a stream. You'd like to know if the rock it's growing on has calcium in it. You can do a test with hydrochloric acid, which you can buy in a hardware store, by the way. And if you put a drop of that on a rock with calcium in it, it bubbles. Where do we have calcium around here? Fall Creek, the limestone quarry down in UC Santa Cruz. There's quite a bit of calcareous rock and limestone. And the lichens and bryophytes too as well really do choose between rocks that have a lot of calcium in them or more acidic rocks like granites. The colors of lichens can be quite variable, but they're always noted in lichen descriptions, and so you want to be looking at colors. Now, colors can be very, very subtle, and it's important to assess color only when they're dry, because the cortex, when they're wet, becomes translucent, and the algal layer dominates the color of the thallus. But look at these two. They're growing right next to each other on the same, these were on tree bark. And the one on the left's a little bit more yellow-green. The one on the right's a little bit more blue-green, but the color can be very subtle. And especially if you're seeing only one at a time, it can be sometimes hard to tell them apart. Also, we want to look at the reproductive mode, whether they're using sexual or asexual, and because most lichens use one or the other. So if we're looking at Cerida, for instance, where on the lichen are those Cerida being created? Are they under the lower surface? Are they terminal? Are they on the upper surface? In the older sections, where are the isidia being created? These things are all important, and when you look to try and identify a lichen, you'll be asked those questions. With the sexual reproduction, apothecia are very common. They're the most common form. They can be seen in all the fruticose and foliose and crustose forms. And the colors and the shapes of the rim are all important. And when you collect one of these guys, you have to make thin sections with a razor blade. And this takes practice, but it's with practice you can do a good job. And then you put them on a microscope slide, and then you can see structures like this. And you're looking at this like this is a cross section of an apothecia, and this is the layer that the spores are being formed at, where the asci are. And there, all these different layers have different names, and you talk about what color those layers are or what reaction they have to such and such a reagent. Another reproductive body are called parathecia, and with these, the spores exit through a pore on the top, and they're relatively uncommon. And if you cut a section with one of these, you'd see this cavity where the spores are being created, and here's some spores created, escaping through the pore on the top of the perithecium. Now, a modified form of apothecium is called a lirale or a lirella, and these are what are in what we call the script lichens, and you can see these on almost any tree. They're very common in Santa Cruz County. And they're a different form of apothecia. If you just kind of think of an apothecial disc, and if you folded it, you fold it in the edges, you get these kind of elongated lip-shaped bodies. And in section, they look like this. And here's the layer where the asci and the spores are being created. And here's a couple of spores floating around. Another sexual reproductive form is the pin lichens, and these are much less common, and they're really a lot of fun to find. And in these guys, the ascus dissolves itself before the spores are fully formed, and so you get this little ball on top of a stick that's almost entirely pure spore, and under the microscope, they look like this. So we've spoken a lot about spores, so let's look at some of them. Spores are really important to lichen taxonomy, and we want to look at the shape of the spores, the number of the cells, whether they're brown or whether they're clear. Here's one that is kind of dark blue-gray. It'll eventually turn brown. It has a number of cell walls that are both longitudinal and transverse. Here's a smaller version of that guy. Here's a really interesting looking spore that's super long, and it doesn't have any longitudinal cell walls. It just has transverse cell walls. But these guys can be relatively straight. They can be sickle shaped. They can be even spiral shaped. Okay. Spores can be simply two-celled and clear, like these guys. Or sometimes the two cells are, the spores have two cells, but the cells are very far apart, like this. And sometimes those cells, when they're immature, have very, very interesting shape with very thick cell walls. All these things are things that we look at when we're trying to tell lichens apart. Here's one that is so large that you can see it almost with a naked eye. It's 0.2 of a millimeter or 1 to 0.2 of a millimeter long. And it's a single cell and clear. So color, shape, size, we're measuring these things through the microscope to tell lichens apart. Another tool we use is chemistry. And most lichens produce chemicals that can cause color reactions to various reagents, and the most common ones we use are potassium hydroxide and household bleach. And sometimes the reagent is put on the cortex. Sometimes we scrape the cortex away and we put it on the medulla. And here on the right is a red chemical reaction to bleach in the lichen Flavopunctilia, which is a close lookalike to Flavoparmelia that we'll see on Friday. You can do more sophisticated tests with solvents and thin layer chromatography. That's something I have yet to learn, and hope to learn that so I can learn more lichen chemistry and get better at identifying them. Well, there are books. The one on the left by Stephen Sharnoff is a very beautiful book. It's a paperback, Field Guide to the California Lichens. It has all forms of lichens, fruticose, foliose, and crustose lichens. It has very wonderful pictures, but it has no keys. It does have some descriptions. It has no keys. And I tend to think of the pictures as not necessarily being the most representative of the normal form of a lichen, but they are the prettiest picture the guy could find. And so it sells books a little bit better, but it's still a valuable book. The one on the right is macrolichens of the Pacific Northwest. Macrolichens are the foliose and fruticose lichens, not the crustose ones. They're the ones that most people see and think of as lichens. And this is an excellent book. Even though it's really dedicated to the Pacific Northwest, which is Northern California all the way up to Alaska. Most of the lichens that we have here can be found in that book. It has keys and good descriptions and sometimes line drawings that help separate two close lichens. So those books are both in my library. I have other books for the crustose lichens, and you can spend a lot of money on books if you want to, but these two are the place you would start. So finishing up, let's look at 10 common lichens in Henry Cowell. First, and easily the most common in the park, and I would have to say probably in Santa Cruz County, is this genus called Usnea. It's a fruticose lichen. It's usually yellow-green, although we will probably see a species on Friday that's orange or reddish-orange. It can be shrubby like the one on the left, or it can be pendulous like the one on the right. It has a really cool feature, which is an elastic central cord. No other genus of lichens has this cord. And you can see the cord in the field. You take the strand of Usnea and you pull gently on it. And the cortex and the medulla break away, and this central cord is exposed. And if you pull on it very gently, you can feel that it has a slight elasticity to it. And if you think about it, it would be a very handy item if you're going to be growing on trees and being whipped around by the wind. It'd be very nice to be somewhat elastic. Another very common lichen here is Flavoparmelia. You'll see this a lot on fallen branches on redwood, in the redwood forest. It's foliose, so it has a different upper and lower surface. It's yellow-green on top, and its lower surface is black. The outer lobes are somewhat smooth. They don't have little pock marks. Sometimes there are ceridia in the margins of the central lobes, and its medulla does not have that red C reaction, the bleach reaction, that its close lookalike Flavopunctilia has. Now, here's Parmotrema, which can be easily confused with the last one. It's also foliose, but it has this kind of little blue-green color to it. It's also black below. It also is often serrated on the margins, but it usually has little black hairs on the margins called cilia. You can see them up here and here and here. And that tells it apart from the Flavoparmelia immediately, which does not have those little hairs. There are about three, I believe there are 3 species in the county. I haven't found any more than that, but we'll leave it at the genus level. Here's the 2 of them side by each, and with the Parmotrema on the right and the Flavoparmelia on the left. And you can see there's a color difference, but you can, if you're looking at only one at a time, you could easily make a mistake. Now, here's one you're not going to make a mistake on. This is Ramalina menziesii. It's otherwise known as the lace lichen. It is the California state lichen. You can find it anywhere where there are lots of trees in the state. Not so much in the deserts, but in the Sierra foothills, it's very common. It's abundant in Santa Cruz County, especially near the coast. But its thallus is dissected into a net. And we will definitely see this on Friday. And this is one that you should be easily able to remember. Now, here's one. It's the only crust that we're going to be able to identify in the field. It's cerediate crust. It grows on bark, and you can see the little Ceridia in the inset photo on the left. Some of my friends call this the honey lichen, but the real test to identify this lichen is to take a little pinch of those Ceridia, a really tiny little amount, and in a minute, it has a very, very bitter taste. And so my friends like to torture beginners and call this a honey lichen and say, just taste it, you'll love it. And a chemical test for it is if you put a little bit of the potassium hydroxide on and then put some bleach on, this lichen turns purple. Next is a large foliose colonizer that grows on soil and rock called Peltidra. The ones that we have around here, the photobiont is blue-green, but you can tell Peltidra by turning it over, and it has these veins on the underside, and the shape and color of these veins and the rhizines are important in terms of telling Peltidras apart. So we'll see this growing on trail banks as we get down the pine trail. Hypogymnia is very common, and we'll see some in the chaparral, but it's also seen a lot of times on the branches that fall to the ground. Hypogymnia is one of the few lichens around here that's actually hollow. Sometimes they're ceridiate. I think the form that we are going to see is this Hypogymnia physotes, and you can see ceridia forming underneath these little openings in the end of each lobe, and the lobe breaks open at the end and the ceridia are exposed and they can be distributed. Another very common genus is Cladonia. Cladonia grows in all different forms. You can see it on soil banks, and a lot of times you can see it on redwood bark. The basic modules of Cladonia are these little plates called squamules and these little erect structure called podicia. And the podicia can be club-shaped or cup-shaped or branched. And when they're cup-shaped, they're often quite short, and these are called the pixie cup lichens. They're common on trail banks, and as I said, you can see some growing on redwood bark. Teleschistes is a wonderful lichen to see when you find it. We'll see some on Friday up near the observation tower. It's a fruticose lichen. It's this beautiful bright orange color. It can have either ceridia or apothecia. These orange plates are apothecia, and there's likely 2 species in the park. You can tell it apart from the orange usnea because it does not have a central strand like usnea does. Another common one, and probably many of you seen this without knowing what it is, on fence posts and fence boards, it's Chrysothrix. It's a dust lichen. It's this magnificent fluorescent yellow. We'll see it on fence posts and signposts. We'll also see it on oak bark on Friday. So there's our 10 common lichens. If you want to learn a little bit more about lichens, you can join the California Lichen Society. It's very inexpensive. You can find us on Facebook and Instagram under California Lichens, or on the web at californialichens.org. And we get together and have walks and annual meetings. We love to teach beginners. And eventually you get to know people, nerds like I am, and we geek out on lichens for a few days at a time. But the most important thing, just don't forget, these are really spectacular beings. And I hope you've enjoyed them. I hope that you've learned something, and I'm sure you've got a lot of questions. So let's close it with that.